Animal Development, Chapter 53. Animal development is not cute and cuddly, but the process of how the cute and cuddly get here. Animal development has 4 stages: fertilization, cleavage, blastula, gastrulation, and organogenesis. Fertilization is the fusion of a sperm and an egg, or as the textbook puts it, male and female gametes. Fertilization is usually external for aquatic animals and internal for terrestrial animals. There are 3 events that make up fertilization: sperm penetration, membrane fusion, egg activation, and fusion of the nuclei. In order for membrane fusion to occur, a sperm must penetrate the protective coats of the egg to reach the plasma membrane. The fusion of the membrane and sperm is what starts embryonic development. When the egg is activated, it builds a defense to prevent polyspermy. When multiple sperm fertilize one egg, if more than one sperm were to fuse with the egg, that would lead to polyploidy, having more than 2 sets of chromosomes, which is incompatible with animals but frequently found in plants. The haploid sperm and the egg nucleus fuse and form the diploid nucleus of the zygote. However, the haploid nucleuses do not fuse in mammals. Instead, the 2 nucleus break down and a new nuclear membrane forms around the chromosomes. In some animals, it is possible to artificially activate an egg without the entry of a sperm, simply by pricking the egg membrane. An egg that is activated in this way may go to develop parthenogenetically. A few kinds of amphibians, fish, and reptiles rely entirely on parthenogenetic reproduction in nature. A few kinds of amphibians, fish, and reptiles can reproduce without fertilization. The cleavage and blastula phase. Cleavage is a rapid division of the zygote and does not increase the size of the zygote. Did you know that cleavage patterns are quite diverse, and there are about as many ways to divide up the cytoplasm of an animal egg during cleavage as there are phyla of animals? A blastomere is an individual unspecialized cell in a tightly packed mass of cells. Blastomeres are formed during and by cleavage. A blastula is an early embryo that will typically have a hollow center filled with fluid. A zygote is like the Earth. They both have 2 poles, but egg poles are called animal and vegetal. Cells that form the animal pole will later go on to forming the external tissues, and the cells of the vegetal pole form the internal tissues. The 3rd step in animal development is gastrulation. This forms the 3 primary germ layers: endoderm, mesoderm, ectoderm. The endoderm is the innermost of the 3 primary germ layers of the embryo. It forms the digestive tract and its derivatives. The mesoderm is the middle of the 3 layers and forms bodily tissues and structures such as the tissues, bones, and muscles. The heart is a mesoderm-derived structure in all animals, and it is the first organ to become functional during embryonic development. The ectoderm is the outermost layer. It forms various tissues and structures such as the nervous system, eyes, and ears. How do cells know their location and what to become? The solution to the puzzle is an organizer. The organizer is not something that cleans and straightens, but it is like a GPS device for cells, except it is a cluster of cells on the outside and in between both poles. The organizer releases signals. The other cells diffuse the signals to determine their location. Their location then determines what to become. Organogenesis is the 4th and final step in animal development. Now that the basic body plan has been laid out thanks to gastrulation, the stage is set for organogenesis to occur. Organogenesis has to follow closely behind the end, if not before the end, of gastrulation, because organogenesis occurs by interacting with cells of the germ layers to develop the bodily organs. Simply put, organogenesis is the development of the organs. Like I said earlier, animal development is not the cute and cuddly, but how it gets here. Simply put, it is a miracle of life. And speaking of life, what takes an average of 266 days, 9 months, to be born? A baby. A human zygote undergoes its first cleavage in as little as 30 hours after fertilization. A second week after fertilization takes place, the placenta is formed. One of the hormones released by the placenta is human chorionic gonadotropin, HCG, which was discussed in Chapter 52. This hormone is secreted by the trophoblast cells even before they become the chorion, and is a hormone assayed in pregnancy tests.